during the academic year, and well, a little over, I guess, because it goes through May. But it's basically just a venue to discuss new tools, technologies, and or methodologies that are currently in development, recently developed, or just some neat tool that researchers are using or might find of interest. There's a sign-in sheet going around. If you can just put your name and information on there, it just helps us with the pizza when you know how many people attended to justify the pizza. Of course, if you haven't already, help yourself to pizza, and there's also cookies and brownies over on the printer. So today we have Ya Yang, who is from Ecology and Evolutionary Biology, and I think she's going to give a brief introduction of herself, so I will let her talk from here.
All right. No. Okay. So my name is Ya Yang. You can call me Ya. I'm currently a postdoc at Stephen Smith's lab. So most people in our lab working on computational phylogenetics. These are the people here. So yeah, this is building phylogenetic trees and looking at different methods of doing them is what we're doing. So I occasionally, people work on model organism or like human genetics sometimes ask me the question, so this is a gene that I'm studying. I know the function, but I'm interested in what is the evolutionary history of my favorite gene. So what are the tools that I should use to make a tree? So here are the most commonly used tools. I just list them here as an overview. And basically building gene tree takes 3 basic steps. First, you have is homology inference, finding what are the gene sequence you should include. And the second is multiple sequence alignment. And third one is actually building the tree. It's actually the last step is the least problematic step. So before I get into the methodology details, just a few basic thing about trees, because that's a lot of you don't use trees that much in your daily work. So this is the most common phylogenetic tree representation. Okay, so the root and those are the tips. So one quiz here. There's one tree that are different from the other. Which one is it? You guys, keep quiet. One tree is different from the other. C. Everybody agree? Okay. C. So basically, you look at DE is sister to each other next to C, DE and C. But here, DE is not sister to C, instead sister to DC together, sister to DE. So that's basically how you read a phylogenetic tree. And when you read literatures, there are many different ways of representing phylogenetic trees. They're basically the same idea. It's just this one, you can align the names of your genes or species a little easier. This one is usually seen when you see the coalescence. When they're talking about coalescence, they usually do it in this way. And this way, they usually use in order to draw a bigger tree to save some space in naming the tips. So again, which one is different from the rest? Just to get you used to reading different formats of the tree.
Okay, yeah.
Someone else? A, yes. A is different. Yeah, again, you're basically looking at the branching order. Yeah, great. So another question is, when we're doing homology search, so basically, the first thing I want to stress is homology is basically common ancestry. So it's a Boolean. Either those 2 genes are homologous or not. There's 2 genes can be 90% similar, but it can be 90% homology. There are a lot of literature that talk about the age of the gene. How old is my gene? So and how deep should I go in homology search. This is actually a really great paper when you are wondering how to deal with gene age. Actually, a lot of literature, including some Nature Science paper, does it wrong. A lot of paper, when they deal with how old my gene, they basically take those heuristic orthology search software like OrthoMCL, whatever, and run it through. And if something get break off, they says, this is a new gene origin. That is silly, right? You have to really look at where the gene duplication happened and how you define your gene, right? So it's not an easy question. All right. So I'm gonna go through three most commonly used homology search tools. I'm sure you guys are familiar with BLAST and see this all the time. I just want to remind you that it's a 25-year-old tool. It's fast, but it's heuristic, which means it doesn't guarantee finding everything in a database. And two, it doesn't guarantee the optimal local alignment. It's basically word searching and extension, you know. So always keep in mind that. So, so in my work, I actually most often use swipe for homology search. It stands for Smith-Waterman database searches with intersequence single instruction multiple data parallelization. Basically a speed up of the basic Smith-Waterman local alignment algorithm. It came out pretty recently, but has been widely adopted in a lot of pipelines published afterwards. The nice thing about it is that it uses Smith-Waterman algorithm, so it's an exhaustive search. It guarantees finding everything in your database and guarantees the optimum alignment according to your mismatch and gap penalty, of course. And it's also nice because the input of swipe is your standard BLAST database. And the output, you can use several of the basic standard BLAST output. So it's really easy to insert swipe into your existing pipeline.
How much things, so as you said, you try the software, so how many different results you have got compared to BLAST? Does it differ a lot in this case?
Between BLAST and swipe?
Swipe, yes.
It's quite different. I mean, depend on the complexity of your database, right? Swipe find quite a bit more hits, and it's generally more exhaustive. That's why some of the large-scale orthology inference methods have been switching to swipe. Some of the recent updates. So yeah, it's a very easy pipeline substitution. So you can always try it out. And the benchmark paper claimed that the speed is comparable to BLAST-P, but it's really dependent on your scoring matrix, depending on your data. So I would say it's in the same order of magnitude, to say it this way. It's still quite a bit slower than BLAST-P. And the third major one is Hammer. So it's different from both swipe and BLAST. In Hammer, use the profile alignment to fit hidden Markov model to alignment. So, and then align your query sequence to the profile. So I'm sure some of you are familiar with hidden Markov model. Just for those of you who are new to it, basically, analog is, for example, a person in 10 days, a person walked to work in day 1, 2, 3, 4, and 5, 7, 10, but he drive to work in the rest of the days. So how can you estimate the weather from this data, right? You don't observe the weather. You only observe how this person prefer to go to work. And you know that when the weather is good, he's more likely to walk than drive. So same as alignment. We don't observe sequence substitution. Those are historical events. We only see the outcome of sequence substitution. We see the differences of nucleotides or amino acid. So this basically hidden Markov model is a nice way to model what are the underlying processes and what are the probability switching between one state versus the other. So, so you can estimate protein specific or even protein domain specific insertion, deletion, frequency, and switching rate instead of use a predefined scoring matrix. Actually, predefined scoring matrix is quite a problem for both BLAST and SWIPE because most of those scoring matrices are pretty old. Most of them at least a decade old. And they were originally from a very small database, a small alignment, some of them just from vertebrate or something. So they could be pretty off for your data set. And Hammer is nice in that it can estimate these parameters on the go. Because it takes the information from entire alignment and make a profile, it's more sensitive than BLAST-P or SWIPE. It can deal with remote homology, which is nice. Actually, Hammer has been already implemented in a lot of pipelines, even if you don't use it that much, but it's in the backend a lot of like pfan database, and a lot of sequence alignment pipelines use Hammer or variation of the similar HMM models. It's also used in actually majority of the multiple sequence alignment. You can do profile alignment, align two profile together and keep each of the profile the same. So yeah, these are the three major homology inference tools we typically use. One thing I want to stress is that, so the use and abuse of the e-value. So all our homology alignment software are local alignment, right? So the output is all about the section that aligns. It has nothing to do with the entire sequence. That's first thing. And also, e-value is basically, for example, you get an e-value of 1, which means you're expecting to get one hit by chance by searching a random sequence, random database from your particular database, which you immediately can see it assume a completely random database, right? And also the e-value is related to your data, the size of your database, and even worse, the size of your query sequence. So if you get bad query or inappropriate query, it tips off the e-value as well. So although all three tools output e-value, but always take the e-value with a grain of salt. So low e-value means it's more likely this is a homolog. But the value of e-value sometimes means nothing. For example, if this is our tree, this is underlying there are 6 species, and you use the sequence from species F as query. So your top hits, what species from A, B, C, or D, or E are more likely to come up with the top hits? Any guess would be your top hits on your BLAST P or whatever?
Like C.
C? Why?
Just guess.
So the root is here. These are the species relationship. So what about the evolutionary distance between these species? Is C more closely related to F or A more closely related to F?
In terms of time distance?
Yeah.
They're all the same.
Yeah, they're all the same. Exactly. So if the substitution rate are exactly the same, right? You're without any other information, you're equally likely to get top hits from any of these 5 species. So that's a problem with E-value, is that your top hits could be any of these. So using the highest one as your ortholog or whatever doesn't mean anything, right? So this is one of the things I see over and over again in literature, that interpret the top hits as the ortholog. You will see that a lot if you pay attention, but this is a big problem in orthology assignment. So one thing as an evolutionary biologist, I scratch my head a lot is orthology is a phylogenetic concept. So basically, orthology are sequences that split from each other when speciation happens, right? So when you have a gene family like this, you have a gene duplication. You have 2 copies, and these are all speciation events. So you will see species A have this copy and this copy. So they are paralogs, and these are all orthologs. So the right way to do ortholog is by building a tree, right? There are many scenarios why your top hits, no matter how rigorous your method is using hammer or swipe, your top E-value may not be your ortholog. For example, using BLAST, especially using BLAST, you can have non-optimal alignment or just miss that sequence. Or you can have model misspecification. No matter how sophisticated your model is, it might not fit your data. The model can come from vertebrate, but your data is fungus or virus, right? And you could have substitution rate heterogeneity. I know that in mammal, substitution rate is highly correlated with generation time and metabolism. So substitution rate heterogeneity is everywhere. And also, like, different copy of genes can sometimes have different selection pressures, so things can throw off your E-value. And you could have incomplete sequence from misannotated genome sometimes. You actually see it a lot, surprisingly. A splice variance, and each splice variance can give you different E-value. Or you can have structural changes. The conserved motif might repeat 5 times, 7 times, and domain shuffling. All those things can throw off your E-values. And then genome duplication. Although people traditionally thought genome duplication is more of a plant phenomenon, but now with more and more genome come out, there are genome duplication in vertebrate, and especially a lot like fishes. And so it is, although you can use you can align genome and use synteny to do your orthology. But when you have genome duplication, you have 2 copies that are both, in theory, orthologous to your ancestral copy. So orthology is really a relative term. You better do it from a tree. So there are a lot of homology, orthology software or methods out there. Like reciprocal best hit is the worst one. There are a lot of graph-based orthology inference methods like OrthoMCL. Those are commonly used, but they are heuristics for large-scale orthology inference, which is fine, they're heuristics. But if you're interested in your favorite genes, you should use the slow and the hard way, so that you can get a really good picture of the pattern that you're interested in. So yeah, so next step, in order to build this kind of tree to look at orthology and gene family evolution, you have to build a tree. And in order to build a tree, you need an alignment. So math is my personal favorite alignment software I'm using. It's coded in my pipeline. It's always the first thing to go to. There are a few reasons. First, it's very versatile. It has options that you can do. All the major algorithms are coded in math. You can pick what algorithm do you use, depend on whether you're aligning distant related or not, or how big of a alignment it is. Like 1,000 or more than 1,000 sequences, you will use different algorithms. So it incorporates progressive alignment, which is the oldest alignment method. That's basically what classical W does. That's basically what I was taught when I was undergrad. So it basically do a distant matrix and find the two most closely related sequences and do global pairwise alignment, and then continue doing. So basically, if you have n sequences, you do n-1 pair. So it's it's scaled linearly, which is very fast. You can deal with very large alignment. And then you can, one of the slower, oh, go ahead.
What's the difference from this math to muscle?
Math actually does a lot of things that muscle do as well. But math is faster and it has more options. So I used to use muscle a while ago, but I think the past couple of years I just use math. Sometimes it's a little bit convenience as well. Math actually borrow a lot of things from classical W, muscle, and T-coffee. So, and even nowadays at classical Omega copy a lot of things from math as well. It's the next generation of classical W. So it's a lot of times it's just different implementation of very similar algorithm. Yeah, and math is very fast, and it multi-threads really well. And I compile in the school cluster. They have the Intel compiler, which speed up even more, like a few times more. So yeah, sometimes it's just out of convenience, like accuracy-wise, like different benchmarks. Sometimes they have conflicting results, but yeah, it's fast and it's convenient and it very easily implemented into pipeline. And also, as you can see, it's a very actively updated software, currently at version 7. So it does progressive alignment. Muscle does iterative alignment as well. So basically, just divide alignment and realign each, divide and conquer, and put them together, and do it over and over again until no better alignment can be found. They also does profile alignment. So if you have an existing alignment that is good and you want to align like next-gen reads, short reads into your existing alignment, math does that. Actually, some of the more recent updates of math is to be compatible with next-gen data types. So yeah, this is my daily tool. But as you can see, math tend to over-align. What does that mean? Means look at these areas that are relatively fast evolving. Math sometimes don't know what to do. And same does classical W or muscle. All these alignment tools have problem with these fast evolving region. So another tool that's really nice is Prank. It's a relatively recent tool. It does, first, you can give it an input tree or alignment, but it will do its own alignment. It will do alignment, make a tree, and re-estimate alignment that take evolutionary events into account. So which you can see that this is the alignment using classical W. This one is using Prank. As you can see in progressive alignment, one of the problems is that when it sees these variable region, it just leave it out like that, or just do whatever that maximum is alignment score, and whatever it can't align, just leave as gap, right? And also progressive alignment, because it's progressive, whatever mistake that happened in early stages will carry on. So you would have more likely to have some of the weird stuff, weird gap. Versus Prank, it take each gap, the generation and closing of gap as distinctive evolutionary events that will be inherited along the tree to all those descendants. So it is tree-informed alignment program. In benchmark studies, Prank usually outperform math or muscle or classical in the accuracy, but Prank only works well with relatively closely related sequences. When you get more distantly related sequences, it will just get really gappy alignment that doesn't make sense at all. Like just really, really large. Sometimes it will lead to a group of sequences that doesn't overlap in any column. It's really odd, even if they are remotely homologous. So yeah, and it's one problem with Prank is that it's slow. There's no parallel. It's single thread, and yeah, and because it does those progressive thing, I wouldn't use it for more than 1,000 sequences. So, which means it actually works well if you have closely related sequences and not too big. You can try it out. Yeah. So, another tool that's used a completely different method is FSA, Fast Statistical Alignment. One of the major difference is between FSA versus most other alignment methods is that FSA doesn't use a guide tree. Even math, it calculates a really rough tree to start with. It doesn't so math doesn't iteratively refine with a guide tree, but it starts with a guide tree. And of course, Prank do the iterative refinement of both the tree and alignment. FSA doesn't use that. It uses pairwise Hindmarkov model, and it gives you the confidence of different regions of the alignment. It works with, I would say, less than 2,000 sequences, so work with most of the alignment you're going to deal with. Sometimes they get quite different results, depend on how distantly related your sequences are. It certainly works with more distantly related sequences than Prank. So sometimes Prank just doesn't work, and FSA is a nice tool to have. So yeah, I actually spent most of my time talking about homology, inference, and sequence alignment. The reason is that the tree inference step is actually the easier step and more mature step. The two previous steps are more problematic in a lot of literatures. People misuse them quite a bit. So as for tree inference, there are a lot of tree inference software out there, but my personal favorite, or our lab favorite, is RexML, right? So RexML, since it came out, it's basically revolutionized evolutionary biology, how we make tree and how big of a tree and how accurate we can calculate trees. It's relatively fast, and it's very accurate, and it's multi-threading. It works very well, and it has those different. If you look at, it has like 20 different makefile if you download it, and different chips use different makefile and different architecture of like parallelism, you use different makefile. It's quite fun to play with, and you can use Intel compiler, which further make it a lot faster. We do a lot of like benchmark because we use this so much. We benchmark this, and yeah, it's really nice. It also and basically it's made in the Alexis group, and the person who wrote this is master of cutting corners in programming stuff. So he speed up things quite a bit. And you probably use some of other maximum likelihood calculation software like in Pulp or Phimo or Mega, but this one is a lot faster than the predecessors. And the even faster maximum likelihood tree building tool is FastTree. I usually switch to FastTree when it's over 1,000 tips, and it does some further cutting corner stuff than even the RexML, and of course it's quick and dirty. But when you compare the tree from FastTree versus RexML, it's usually the well-supported versions are the same. It's usually when you have quick diversification and lack of information, those nodes. FastTree is easy to stuck at local optimum in its hill climbing. So these are the two tools I use the most. And then there's Mr. Bayes based on its Bayesian inference instead of maximum likelihood. So when you do a maximum likelihood at these two, you get one single maximum likelihood tree that maximize the likelihood score. But when you do Bayesian, you get a posterior distribution of tens or hundreds of thousands of trees that represent the uncertainty in a tree estimation, which can be really nice because knowing the uncertainty in the tree. Unfortunately, it doesn't deal with very large trees because they do this propose a new topology and then maximize everything and then walk through the tree space. It only deals with a few hundred species, and sometimes it just, when tree get really large and complex, the chains doesn't converge. So it's not as fast as the other methods, but when we're really interested in the tree uncertainty, we will use Mr. Bayes. And there's another Bayesian method, it's Beast. Beast is really nice because it does molecular dating. So when you have any data set that you either know the dates of the tip or dates of any of the internal nodes, Beast is a superior method than Mr. Bayes. This is a recent paper of Ebola outbreak. Beast is very commonly used in infectious disease research. It's because when you have infectious disease samples, you often have the dates because you know when the sample was collected, right? And sometimes you have the geographic distribution. For example, HIV samples, you know from which person this population of virus was diverged. So when you have these additional information, it can constrain a lot of the nodes to be at certain state or at certain time point in order to infer the state and time point of other nodes, like when is it started, Ebola, in the case of Ebola. So this is the overview from 1975 to current of the evolution of Ebola. And this is a zoom in of year of 2014. And you can see like the tip. One of the interesting things to look at infectious disease trees, they're different from typical gene trees because they're tips that finishes at a certain time point. That's your sampling time point, right? Basically that's when you sequence the sequence that you got. They each carry a time point information. So Beast is a nice tool to incorporate these additional information. Yeah. So there are a few tips for tree inference. Sometimes people make wrong assumptions that sometimes I found. First is that it's more important to have good alignment and correct homolog and ortholog group than making these are more critical step than the final tree step. A lot of times people fight over its tree should be this way, not that way, but they don't go back to look at their initial steps. And sometimes after you get the tree, then you get your correct ortholog group, and then you reduce to the subtree, a smaller set of ortholog, then you redo the whole process to refine your homolog group and the focal clade on your gene family. And also picking the correct model is important, especially when you have some of the nodes are lack of information. Sometimes one person get one topology, the other person get the other one. It's because they have different models. As I said, a lot of the models are old and from a small database, and sometimes it might make sense to estimate your own substitution model based on your group of species. And if you deal with DNA, don't ever do model tests, or don't, I wouldn't say ever, but usually model tests is not required because people used to do model tests a lot previously because computers were slow back then. It sometimes just makes sense to use a simpler model than use a more parameter-rich model. But nowadays, with a better computer, most of the time it's just use the GTR, general time reversible. It is one of the more parameter-rich model. So, but when you look at really deep relationships, like hundreds of millions a year, that's when general time reversible would break, or like they evolve really fast, because general time reversible, the only assumption is that the probability of switch between A to T and T to A is the same. But over a long period of time, those would change. So you might use an even more general model. And next is use maximum likelihood or Bayesian. Don't use neighborhood joining or UPGMA. Again, neighborhood joining or UPGMA are clustering methods that are used when computers are slow. Because the first step of both NJ and UPGMA is to make pairwise distance and erase all the information in your base pair or amino acid and use those distance to make a tree. They lose a lot of information, so don't use NJ or that. Just use maximum likelihood. Your computer should be able to handle it, even on a laptop. The next thing is tree uncertainty. So there are two major ways of dealing with tree, looking at tree uncertainty. In maximum likelihood framework, people usually use non-parametric bootstrap, which means people subsample the columns in your alignment and make a tree and repeat this 100, 200, 500 times and see how often they agree with each other. But it's basically, it's not a measurement of how much confidence you put in your tree. It's measurement of the consistency of your data, right? So basically, if you have your whole genome, your bootstrap is always going to be 100% because your data is super consistent. It doesn't mean your tree is correct. You might specify the wrong model, but get 100% bootstrap all the time. So the interpretation of the numbers on your tree is very important. And then posterior distribution is actually the confidence given of your tree given your model. So again, when you get 100%, it just means given your model. It doesn't say anything about like how true the tree is. You have to scrutinize some of the earlier steps. And then it's a nice tool for visualizing your tree and check whether everything makes sense. The big tree is a nice tool we use daily. You can color your benches and basically give you a publication quality figures using fig tree is kind of nice. So phylogenetics methods are so powerful that you can not only build trees using sequences, you can also build trees of languages. Right? You can even have fossil languages of dead languages and put in a tree, see how they relate to other languages. But building any tree using like cultural things, you have to worry about borrowed words and stuff. There is a nice TED talk by Mark Pigo about his work on tree of languages. You can also build tree of dead animals using morphological characters. You can even build tree of imaginary creatures. This is actually a t-shirt you can buy online. So yeah, the Oriental dragon is the most distantly related, I think. Yeah. Yeah. So that's. Am I out of time, or I have
Nope.
more slides, but
You got 10 minutes.
You have about 10 minutes.
all right. So I'm gonna give a little bit spiel of my own work. How am I applying these methods? So yeah, I'm an evolutionary biologist. I'm interested in evolution of species beyond the few model organism species. I want to have the freedom to look at whatever species I'm interested in and compare them to model organism. So I reconstruct species relationship using genome and transcriptome. I sequence transcriptome from non-model organism and do de novo RNA assembly. It's basically I use that as a genome reduction method. I also look at differential gene expression, but the first step is to get the coding sequences. You would think this is an error prone and you'll have a lot of missing data, but actually for most plants, there are about 5,000 genes that are consecutively expressed. You get nice assembly and you get at least 5,000 gene trees, at least, by comparing fairly distantly related species. And the fun part is when they disagree with each other. So what's causing these disagreement? And whether those are because of error or lack of information, or they actually mean something adaptive. So that is what I'm studying. So yeah, previous work, usually this is a matrix occupancy. Basically, you rank all the ortholog from include 100% of the species versus include versus a few of the species that you're interested in. Previous method, because their orthology and homology assignment problem, they use like ortho MCL, reciprocal best hit. You lose most of your information. You get a lot of things wrong. So in the end, after your filtering step, you usually get a few genes that have all of the species. And most of the genes have not much information, not many species in it. But I started out, did a benchmark studies, look at species with genome and also have high coverage transcriptome. And usually around 50% of total gene in the genome, usually housekeeping gene, are pretty accurately assembled using de novo method, which is counterintuitive for a lot of people for de novo assembly. And I also benchmarked the error rate and chimera rate. It's there, but it's tolerably low, and you can use some other filtering step to get them out in the later stages. So really, your ortholog group should be something like that. You have some housekeeping genes and then tail off to some differentially expressed genes. So this is I do ortholog inference, use this kind of framework. And because the first step is large scale homology inference. I start with the all-by-all blast and the Markov clustering methods. Basically, this is my all-by-all blast results. Usually when you're doing all-by-all, it tolerate a little bit of missing stuff because all you want is to get a cluster of highly connected homologs. So you can fine-tune the parameter you use to get the clusters, but I tune it in a way to get relatively crude cluster, relatively big, and this is a rendering of, you can see some of the big gene families get pretty big clusters in the middle, but most of them are nicely scattered, usually much smaller gene families. So, and then next step, I build a phylogeny. Usually use math and RexML. You build a tree like this. This is a homolog tree, and I have my out groups and in group. Out groups are the species that have genome sequences that are distantly related to my focal group, and in groups are my focal group, so that I can root the tree and extract the in group that are separated by the out group. So that, and this is my extracted rooted tree. And sometimes you see gene duplication like this. And I'm interested in both homolog and ortholog from my focal group. And this is informative by itself, but the next step I do is I cut apart the ortholog groups. And because this is a rooted tree, so I know this is a gene duplication event. So my study plants are some extremophytes. Plants are adapted to extreme environment. Those include cacti, and those are Venus flytrap. They're odd-looking plants that how they become like that. And one of them, sugar beet, is also in my group. It's nice because sugar beet has full genome sequence, so it's nice to always have something to scaffold your data. I reconstructed the species. You don't have to read, but I ended up getting more than 10,000 homologs and close to 20,000 orthologs that have a fair bit of taxon representation. And I made a species tree with more than 1,000 genes using RexML. Yeah, I also look at the discordance among these gene trees, which is, there's quite a bit of discordance. There are allopolyploidy in quite a few places in this group. And I look at and map the pattern of gene duplication. So what are the phylogenetic location of the gene duplication? So they turn out to be, there are six phylogenetic hotspots of gene duplication. And 3 of them are known genome duplications, and this one is in Arabidopsis. These are 2 successive genome duplication. That's why it's a lot of genes get duplicated. The diameter of those bubbles are proportional to the proportion of genes that I can detect gene duplication. And also, I did synonymous distance analyses and confirmed that these 3 are previous unknown genome duplication events. I feel like if we can find genome duplication just using transcriptome data, these are from genome data. These are all from transcriptome data. The actually pattern is pretty consistent. So yeah, so maybe there's more that people just don't know. We don't have genome data, but we don't need genome data to see those deep genome duplications. And you can't just count chromosome number because they're very ancient. And I also look at the functional location of the genes that went through the most gene duplication. These are transcriptome data, so they are missing. They're not necessarily, underestimate gene duplication, right? Because you have missing data. But my assumption is that if you see gene duplication in your transcriptome and they're co-expressed in high level across the whole group, this is expensive. This could have some implication for adaptation. So one of the most highly duplicated and co-expressed gene is the phytochrome P450 gene. Actually, phytochrome P450 is a gene that's involving a lot of adaptations. For example, in insects, a differential expression of member P450 is associated with like whether you're a queen or you're a worker. This is quite an interesting gene. This one of the duplicated copy of this gene has become a key enzyme in a Bayesian synthesis pathway, which make this bright red color of your beet. Bayesian is actually really interesting. It shares some initial synthesis steps with melanin, which is give your hair color. So both Bayesian and melanin, the first couple of steps basically terracinase, from terracin and terracinase to DOTA, and those steps are shared. But Bayesian is only in plants. It only occurs in my study group. Bayesian is also occur in fungus, which is odd. So yeah, this is a very interesting novel pigment synthesis pathway that's found in beets. Yeah, so next time you see beet, the red color is different from other like red flower you've seen like strawberry. These are different. This is more closely related to your hair color. So yeah, I also look at the evolutionary rate of substitution. We know that in animals that life history has a lot to do with your substitution rate, but in plants, it's a different. We have a lot of perennial plants. So the generation time is a little bit less obvious than in animals, but we can see that we compare the substitution rate in woody versus herbaceous plants. We find that it's a really striking pattern that substitution rate in herbaceous plants is up to 3 times as fast as woody plants. So that's one of the, so you can look at evolution rate using your homologs, right? So yeah, so this is my last one. So this is what I'm currently doing. I'm sampling a densely sampled transcriptome targeting key evolution of interesting traits to look at the evolution of gene family. And we're developing traits for visualizing phylogenetic signal and conflict among homologs. And yeah, so we're dealing with large, incomplete, and noisy data sets. So better tools, informatic tools, we're working on those. So yeah, that's my presentation. Thanks. Questions? Or any additional comments from you guys?
I have a question about with your maximum likelihood trees, where you get a single most likely tree. What are you doing to kind of check that there's not something that's pretty close to that? You know, just like some little thing that might have been different. Like, you know, if you did parsimony, you might find multiple equally parsimonious trees, or take a step back and
do that too. You can run XML multiple times. You can force it to have different starting tree. Yeah, people do that quite often, like do it 3 times and see whether they converge.
Thank you.
Thanks. For you.